Um, like before I start, I want to ask you, how's MOL going so far with you guys? You can type in the chat. Is my voice clear? Okay, thank you for responding, Ahmed. I thought maybe my voice is not reaching. How you will feel it's difficult in the beginning, but it will get easier with time. You just need to learn the best method of studying which suits you. So I'm gonna start now. Uh, you can you can stop me whenever you want, whenever you have questions. Don't hesitate. There's no stupid questions because remember when I was first year, I used to I used to think, oh, this is a silly question, or the doctor is talking, I'm not gonna interrupt. Don't think anything. Whenever you have question or anything, you can just stop me. طيب يلا. Bismillah. Okay. So before I start talking about the reaction which is going in the Krebs cycle, before I start talking about all the details, I want to give you an overview. I want you to understand why is the Krebs cycle happening? Because why is more important than the question what. Like, what is the purpose of the Krebs cycle? So, Krebs cycle will not give us the energy directly, okay? Krebs cycle will give us resources. And this is very important to understand. So what do we mean by energy? Um, yes, ATP. Actually, I can't see them. Um, I can't see the questions on the chat, so I have to uh, go back and forth. You guys can open the mic if you want. Like, um, so what I just said is, so we are not really making energy, okay? We're making a very few ATP, and this is not counted. But the main thing that we are doing in Krebs cycle is that we are making the resources. طيب, the resources that we can use it later on to make to generate ATP, to generate energy. What do we mean by resources? By resources, I mean the electron atoms, okay? Because when we're talking on the cellular level, we need to have these electrons. We're gonna utilize them with oxygen to have our ATP. طيب. So in the Krebs cycle will give us the electrons that we need, the resources. Like how is the Krebs cycle gonna present them? Electrons, and they cannot be free, صح? Electrons cannot just travel around freely. We need something called NADH and FADH2. Okay, NADH and FADH2 are energy carriers. Okay, they are not the energy itself, they are the energy carrier. So with the Krebs cycle, we are hoping to make so many NADH and so many FADH. In other words, we're trying to make so many energy carrier molecules, which we can use further on in different reactions to generate our ATP. Is it clear? I have to... Okay. Um, now, Bismillah. So, Krebs cycle is generating energy, but it's indirectly. تمام? It's generating the energy carrier molecule, which we can use it later on to generate our ATPs. طيب. So I'm sure that you have already taken glycolysis, and even if you haven't, it's fine because I'll try to go through them all. So we have three things in our cells, right? When we are trying to make energy. We have glycolysis, Krebs, and then electron transport chain. So let's start with glycolysis. So we are going to start with what? So what's going to be the first step of the glycolysis? To eat something, صح? You need to eat something, so you get like we always say, the food is the source of the energy, right? So we need to eat, and then we will get our glucose. The body will absorb the food, and then with and it will get glucose. Now in glycolysis, what happened? We broke the glucose into two pyruvates, and we got 2 ATP and 2 NADH. And this is it's very few. If you compare it to Krebs cycle and electron transport chain, it's a very few energy because we did not have oxygen, صح? In the Krebs cycle and electron transport chain, we have oxygen. So we can generate more energy, more ATPs. Now, what's going to happen in the Krebs? You can think of it as a continuation of glycolysis. So this pyruvate will go to the Krebs cycle, okay? And through this cycle, Krebs cycle, we will get so many FADH and NADH. And we just said NADH and FADH are energy carriers. They're electron carrier. Now, the third step, and if you look at the name, it's called electron transport chain, صح? So you can understand that we need electron here. This is the final step to make ATPs. So what's going to happen? This NADH and FADH will go here. And then it will go through these proteins, which you will take it in next lecture, electron transport chain. And here we're going to get the ATPs, the main source of energy. طيب. So Krebs cycle was preparing the NADH and FADH too, because we need them in the electron transport chain to have the ATPs. Okay, is it clear? I'm sorry, I have to escape to look at the chat. Is it clear? Thank you, Ahmed. طيب. طيب. Now I want to talk about NADH and FADH. Like, I want you to get more familiar with it. After that, we can enter the Krebs cycle. Like, to understand that, you have to first understand the oxidation reduction reaction. And I'm sure that you have taken it in chemistry classes in high school. And I hope you remember that. Like, so we have two reactions, which is called oxidation and reduction. In oxidation, we are losing electrons. In reduction, we're gaining electrons. So if you look here to this example, you will see that this substrate have electron, صح? And then we have got this NAD+. So what's going to happen? The substrate is going to give the electron to the NAD+. When the substrate will lose the electron, or we can also use the word hydrogen, because hydrogen has also one electron, صح? In the outer membrane, if you remember. So we will call this substrate that it has been oxidized. Okay, now, when the NAD+ will accept the electrons, it will become NADH. Okay, so NADH is the reduced form. So oxidation and reduction is opposite. In oxidation, we're losing electron. So for example, this substrate, which had electron, it went to NAD+ and it become NADH. And the opposite will be reduction, where we're giving the electron away to someone. So NADH is being reduced, and it's gonna convert into NAD+ again. And another way to think about them is just like these energy trucks, if you see them. So we have a NAD and we have FAD. And I always imagine them as energy trucks. They're roaming all around the mitochondria, and they're providing the energy whoever needs it, and they're taking away the energy whoever doesn't need it, or whoever is donating. Tamam? Now, what's the difference between NAD and FAD? NAD and FAD are same. Okay, they're same. They both are taking. They both have the tendency to take the electrons or to take the hydrogen atom, and they have the tendency to give it away. But the difference is FAD can accept more electrons compared to NAD. So FAD is like stronger, compared to the NAD. Now the enzyme, which does this oxidation, reduction, and reaction, most of the time, we call them dehydrogenase. So whenever you see the word dehydrogenase, you have to understand that there is an oxidation reduction reaction happening. Okay, so that's it. I hope it's clear. Now, if you remember, the glycolysis is occurring in the cytoplasm right here, but the Krebs cycle and electron transport chain is going to occur in the mitochondria. Now, our mitochondria have two, like the component of the mitochondria, it has got two areas. The one inside is called matrix, and these foldings are called cristae. Now we are going to focus on matrix because the Krebs cycle is going to happen inside the matrix of the mitochondria. Like, now we have got our pyruvate and it wants to enter the mitochondria. So we have got two channels, like two protein, which will help the pyruvate to go inside. So for the outside membrane, we have a porin, okay? The full name is voltage-gated porin channel, but you can also only say porin. So in order to pass the outer membrane, the pyruvate has to go through this voltage-gated porin channel. And in order to pass the inner membrane, the pyruvate has to go through the MCP, which is short form for mitochondrial pyruvate carrier. It's a very easy name. The name tells you the function. It's a mitochondrial pyruvate carrier. That's all what you need to know about the pyruvate transportation. Okay. Now, this cycle has three names. And the reason why you need to know these three names is because in the exam, they might use any of the name and you would get it confused. So you need to keep in mind that whenever they're saying Krebs, they mean citric, and at the same time, they also mean the TCA. So why is it called Krebs? It's called Krebs because the person who discovered the Krebs cycle, this genius, mashallah, his name was Hans Krebs, and he was German. I don't know, but I don't know. Whenever I see anything in biochemistry and psychology, they're always Germans, mashallah. Okay. The second name, citric, it comes from the fact that citric acid is the first intermediate of the Krebs cycle. That's why, we honor the citric acid because it's the first intermediate. We can also call it the citric acid cycle. Like the third one, which is TCA, a short form of tricarboxylic acid, this comes from the structure of the citric acid cycle. Now remember, you don't need to know anything about the structure. You can just ignore it, just to skip it. But I'm putting it here for you just to understand why is it called tricarboxylic acid, just to fulfill the curiosity. Now, if you look at the citric acid structure here, you will see that we have got three carboxylic group. That's why it's called TCA, tricarboxylic acid. So yeah, you have to keep in mind that all the three have the same meaning. They are the same thing, basically. Okay. Now, before we start the Krebs cycle, we need our acetyl-CoA. Tamam, before we start the Krebs cycle, we need to have or to generate acetyl-CoA. Okay, so what we have done so far is, before Krebs cycle, it was glycolysis, right? So we had our glucose, we broke it down into two pyruvates. Now we're going to take one of the pyruvate, and we have to convert it into acetyl-CoA. So this is a pyruvate, and we're going to convert this pyruvate into acetyl-CoA through an enzyme called pyruvate dehydrogenase complex. This is a very important step. And the reason why it's very important, because this is the enzyme which is going to decide the fate of Krebs cycle. Whenever this is the one which is going to inhibit or activate the cycle. That's why it's very important for you to remember it. Also, as you can see, it's a complex. So it's not just one enzyme. It's different enzymes working together. We have E1, E2, E3, and alhamdulillah, luckily, we don't need to know the details. We just need to know that this enzyme exists, and we need to know the cofactors. So I'll tell you later why did I put this anime of a nerd person. But for now, this is exactly how I imagine this enzyme. A very huge, a very complex enzyme doing so many things, very important. And this is the one which is gonna decide the fate of Krebs cycle, if it's gonna, if you're gonna activate it or if you're gonna inhibit it. Okay. Now there's one thing. Maybe you wouldn't like it, but you have to remember it, that you need to remember the 5 cofactors which are needed for pyruvate dehydrogenase complex. Actually, as I have said, if you're studying only biochemistry, not the one for medical student, you will see it's more complicated. So actually, you have to thank God that you only have to remember these 5 cofactors, and you can do it randomly, and you don't even need to remember it by order. Okay, so the mnemonic is the sentence which helps us, the random words that we make, which will help us to memorize something. That's what we mean by mnemonic. So the mnemonic to help us remember these cofactors are called the lovely coenzyme for the nerds. That's why I put this picture. And it's not from an enemy, by the way. It's just a random. It's generated by AI. طيب. If you look at the cofactors that we need for the pyruvate dehydrogenase complex, you will notice that all of them are vitamin. So we have vitamin B1, B2, B3, B5, and the last one is just like lipoic acid. Okay, you will see that B1 is thiamine. Always remember, B1 is always thiamine. B2 is fat, and we have already been introduced to fat, صح? So we know it, خلاص. B3 is NAD. This is also not new. We know it, خلاص. B5, and this might be new for you, is CoA, which is coming from pantothenic acid. Okay, so these are the 5 cofactors that are needed for our pyruvate dehydrogenase complex. طيب. Now my question is, are we ready for Krebs cycle? I can't see the chat. Okay. So as you can see in the slides, the first intermediate for Krebs cycle is citrate. So we need to have citrate, and after that we can start the citric acid cycle. That's why it's called citric acid cycle. So now the second step is going to be converting the acetyl-CoA into citrate. How are we going to do that? So acetyl-CoA will bring a friend for help. We're going to bring oxaloacetate and oxaloacetate with acetyl-CoA. And of course, we need an enzyme, which is citrate synthase here. This will give us citrate, tamam? And citrus, this is the first intermediate of Krebs cycle. خلاص? Okay. Now this is just a mnemonic, which will help you to memorize the Krebs cycle intermediates. So the mnemonic says, please, can I keep selling seashells for money, officer? Please, can I keep selling seashells for money, officer? P is the pyruvate. C is citrate. And you will see what other ones are. I don't want to spoil it for you for now. طيب. So, since we have generated our citrate and memorized the mnemonic, are you ready to start the Krebs cycle?
Yes.
Okay, just give me a second. No, there's no way that I can see the chat and this together. No, it's either this or that. Okay. طيب. Never mind. Now, Bismillah, we will start the Krebs cycle. Okay, so let's start with the things that we have talked so far. We said that we had to generate citrate from acetyl-CoA and oxaloacetate, صح? And we said that the acetyl-CoA, from where did we get it? From pyruvate. We said we had our pyruvates, and then we had our enzyme pyruvate dehydrogenase complex, which we said it's a very important enzyme which decides the fate of the Krebs cycle. And now, since we have our citrate, we can start. So before that, remember that we also have FAD and NAD. Okay, so FAD and NAD, you can just as I said, you can imagine them as like energy truck. They're gonna provide or take away the energy, and you will see how they will be useful in reactions. Like, so the first step, I know number 1 is on citrate, but since we have already talked about, for me, this will be number 1. Okay, so the first step after generating citrate is generating isocitrate. This is a very useless step. You know why? Because we have not used any energy. We have not used any energy, and we have not generated any energy. What we basically did is we just converted citrate to its isomer, isocitrate, you know. So yeah, it's like pretty useless. I mean, pretty useless for you when you study, okay, not for the Krebs cycle. Now, the second step is converting the isocitrate into something you will see right now. The second step will be held by an enzyme called isocitrate dehydrogenase. And what did we say? What does dehydrogenase mean? We said we use the enzyme dehydrogenase when we want to talk about oxidation and reduction reaction, right? Tamam. So what's gonna happen? You have this isocitrate, okay, and it's gonna donate its electron, or it's gonna donate its hydrogen atom. Like when it will donate it, who will come and take it from the isocitrate? It's not gonna float, alone in the mitochondria. Something has to come and take it. This will be NAD. Okay, this will be NAD. So NAD will come. Bismillah. Yeah. So NAD will come and it will take one hydrogen from isocitrate and convert it into alpha ketoglutarate, and the NAD become NADH. So this is like in this reaction, we actually generated something. In the citrate to isocitrate, we generated nothing and it was like useless. But here, from isocitrate to alpha ketoglutarate, we have actually generated NADH, our first NADH. And that's why this one, and I have put a little star for that reason, this is a rate-limiting enzyme. Okay, this is a rate-limiting enzyme of the Krebs cycle. What you will also notice is NADH is not the only byproduct, but we also got some carbon dioxide. So, and you will notice in citrate that the citrate was 6 carbon atom. Isocitrate also, if we count here, it was 6. As we said, we haven't changed anything. But in alpha ketoglutarate, because we have removed a carbon dioxide along the hydrogen, you will see that this one will have 5 carbon atoms. So we have decreased the number of carbon atoms. So whenever you will see the reaction has taken away a carbon dioxide, obviously you will have less number. Okay, now the reaction that comes after this, it's gonna be again a dehydrogenase, alpha ketoglutarate dehydrogenase. And what did we say? Whenever we have dehydrogenase, that means a reduction and oxidation happening. And whenever we are having reduction oxidation reaction, we need either NAD, either FAD. صح? Like here, what's going to happen? Same thing. It's going to donate one of its electron, and who's going to come and take it? NAD. صح? And NAD will become NADH. So as you can see here, again, same thing happened. From alpha ketoglutarate to succinyl-CoA, we produce NADH and we produce a carbon dioxide. Okay, now the difference between isocitrate dehydrogenase and this one is that this is a rate limiting step. Okay, while this one is not. Another thing which is interesting about alpha ketoglutarate dehydrogenase is that it requires the same 5 enzymes that we have talked about before. We have talked about in pyruvate dehydrogenase. The thiamine, the lipoic, the FAD, NAD, and CoA. Okay, this is the only one which needs, this number of the enzymes. Now the reaction that comes after that is, as you can see, succinyl-CoA synthetase. So it's not dehydrogenase anymore, right? It's synthetase. So what does that mean? I'm gonna tell you. In synthetase, what happened is we're gonna take away phosphate. Tamam? We're gonna take away a phosphate from succinyl-CoA. Now, who's gonna come and take this phosphate from, the phosphate that succinyl-CoA just gave up? Something called GTP. Now you might be wondering, what's GTP? GTP is same as ATP. GTP is same as ATP. The only difference is the place. Some places have ATP more, some places have GTP more. I mean places inside the cell or inside the tissue. Okay, so what happened right here is that succinyl-CoA donated a phosphate. GTP took it and this is GDP. GDP took it and it became GTP, as you can see right here. This is called substrate level phosphorylation. You know why? Phosphorylation because of the phosphate group that I've just talked about. Substrate level because who is donating or who is taking the phosphate? It's the substrate, right? We are not doing it inside electron transport chain or something else. So very easy. So whenever you can have this as an exam question, that which of the enzymes is responsible for the substrate level phosphorylation in Krebs cycle, and the answer will be succinyl-CoA synthetase. Okay. Okay. The reaction after that is succinate dehydrogenase. Again, what did we say dehydrogenase mean? It means reduction and oxidation. So again, succinate is gonna give away an electron, and who's gonna come and take it? This time, it's not the NADH. It's not the NAD. It's not the NAD. This time it will be the FAD. This time it will be the FAD which will take the electrons and become FADH2. So here we have actually generated FADH2. So if in the exam they might ask you which of the reactions generates FAD, you have to remember it's the succinate dehydrogenase, not the succinyl-CoA synthetase. Okay. Like, after that, we have malate, and this is because it's not giving us any energy, so we can just skip this step. The step after that is again it's oxaloacetate dehydrogenase. So what's gonna happen? It's gonna give an electron away. NAD is gonna take it, and we have generated our NADH again. So for the exam, it's good for you to remember like the Krebs cycle in order, but for the exam, you don't really need to know everything. Like in the exam, they wouldn't ask you, please tell me the steps of the Krebs cycle in order. But what they're gonna ask you is these things. They're gonna ask you which is the rate limiting step. So you have to remember it's the isocitrate dehydrogenase. They're gonna ask you which one of them needs the five cofactors, or maybe one of them can be, an MCQ, and then you have to remember it's alpha ketoglutarate dehydrogenase. They might ask you where does the substrate level phosphorylation happens. So you're gonna tell them it's the conversion of succinyl-CoA to succinate. And also you have to remember which one is gonna give you FAD because it's only one. So yeah. Do you have any question? Do you have any questions? Okay, Humaira, I just saw that you were asking me to repeat the part after glycolysis. I can do it now, it's fine. It's okay, you mean me you want me to repeat it, or okay. I'm so sorry because I can't see the chat. Okay. Okay. So I believe that it was okay, خلاص. Okay, so now we can move on. So now we have to understand the relationships. Okay, so now what you have studied so far with me is that Krebs cycle is a closed cycle. I'm sure that this is how you're imagining it. So we have got citrate in the beginning, and then we kept continuing until we reached the citrate again. صح? But this is actually not true because every like intermediate of Krebs cycle can also be involved in other reactions. I know this sounds so confusing, but yeah, that's how, mashallah, everything is so complicated, so complex in the mitochondria. This is how humans are. So the other reaction which are also related to Krebs cycle is gluconeogenesis, fatty acid synthesis, amino acid metabolism, and heme metabolism. Like now, if you see here, and I'm sure this is you guys right now, because this was me when I saw this for the first time. Now so this is the Krebs cycle, so you can ignore everything and just look at the Krebs cycle. Actually, it starts from here, but I've also outlined this part because we have taken it, because you understand what's going on. So what this is trying to tell you is just what I said right now, is that every intermediate can also be involved in different reaction. Okay, for example, you can see that alpha ketoglutarate helped us to make glutamate, okay, which is an amino acid. You have probably also seen here that the oxaloacetate was involved in making serine, glycine, and all these, amino acid. Succinyl-CoA was involved in making porphyrin, which and heme, part of the hemoglobin. Now the question is, do you need to know them for now? Okay, the answer is it depends on your lectures, because all these things which are outside the yellow circle is a different lecture itself. So for example, when you will take the amino acid catabolism and I don't know, the metabolism in general, you will see how that time you can relate it to, okay, this is how it's related to Krebs cycle. But for now, since you have not taken these amino acids, lecture and everything, so yeah, I don't think, like, you just need to understand that they're related, but you don't need to know how and how exactly and the details, unless if you have taken like a different separate lecture about them. Okay. Another thing that I want to tell you is that, okay, I've told you that the glucose is the one which is going to convert into pyruvate, and then it's going to convert into acetyl-CoA, and after that we can enter the Krebs cycle. صح? The glucose is not the only thing that we eat, right? We also eat proteins, we also eat fat, and so can we make energy from them? The answer is yes. Okay, so we can also make energy from amino acid, from the fatty acid. You're not required to know the details for now, but just remember that we can also use them to make acetyl-CoA. Just the same, like if you converted them into acetyl-CoA, acetyl-CoA is the main one, صح? Which is going to enter the Krebs cycle. So if you converted either amino acid or fatty acid into acetyl-CoA, you can start the Krebs cycle, it's fine. But the details, how is it exactly being converted, you're not required to know right now. You're gonna study it in the lecture of the amino acid and in the lecture of the fatty acid. But anyways, glucose is the major resource of the acetyl-CoA for the Krebs cycle. Okay. Now I'm sure you might be wondering, why haven't we talked about how much ATP are we getting, صح? Like, is it 3? Is it 2? Like, I haven't told you how much ATP are we getting from the Krebs cycle, from the NAD and FAD and everything that we got. The answer to this is because they don't know. Everything is under research. So even when you go to the books, different books are going to tell you different numbers. Some books are going to tell you 38, some book is going to go 39. There is no fixed number. So maybe in the future, future students, like after, I don't know, 10, maybe more years, they will have to remember it. But for us, alhamdulillah, because the scientists themselves are unsure, خلاص, we don't need to know it. Okay? Okay. Now the real question is, why do we need to learn Krebs cycle? So probably you might be thinking it's useless. There's so many reactions. There's so many things happening. What am I gonna do with it? Like, what's the purpose of learning all this as a medical doctor? The answer is actually it's not one. There are answers to this question. First of all, the reason is because they're very small. They're very small and they're happening inside the cell. They're overlooked. And most of the time when we can't see something, we consider them difficult and hard. Another thing is because we are living our life in a very easy way, alhamdulillah. Whenever we're hungry, we can go and eat. Whenever we want, whenever we are full, we can just wait. Whenever we are doing exercise, we might, you can get more energy by eating, by drinking water. But think about people who have genetic defects. Think about people who have, for example, one of the enzyme is missing, or one of the enzyme have mutation. What's going to happen to them? How are they going to generate energy? Think about the people who have malabsorption. Now, didn't we say that we need the cofactors in pyruvate dehydrogenase complex? Now, imagine if someone isn't having enough nutrition, if someone isn't having vitamins, what's going to happen to them? You as a doctor, you need to know what's going to happen. You need to know the pathophysiology, and you need to know mainly how to solve this thing. Also, there are so many drugs, there are so many poisons, and there are so many, not so many, alcohol, which will impair this Krebs cycle, and you need to understand why. Also, whenever you have a problem in the Krebs cycle, whenever the body is unable to use, the oxidative phosphorylation, what's going to happen? The glucose will go into other way. It will be converted into lactate. And whenever you have an elevation of blood lactate, it's not a good thing clinically, and you have to intervene it as soon as possible. So these are the reasons. Whenever you're thinking the Krebs cycle is not like it's useless, you have to always look to this. Like, now to be more specific, if you remember, we have talked about 5 cofactors, صح? We have talked about B1, B2, B5, B3, and lipoic acid. Now, these are important. You have to know the diseases that are related to it. What's going to happen if you have deficiency in any of these things? Let's start with B1, which is thiamine pyrophosphate. Okay. So for thiamine pyrophosphate, you have to remember 2 diseases, beriberi and Wernicke-Korsav syndrome. Beriberi actually comes from a Sri Lankan word which means weakness. So if someone is from Sri Lanka, they would understand the meaning. And this one is named after a German scientist, of course. So now, this disease, most of the time, it happens in people who abuse alcohol and the people who are malnourished, people who are homeless, people who are from like poor places, These are the people who are mostly affected by the thiamine deficiency. And the organs which are mostly affected is heart and nerve. Like the two of the most important part of the body is affected by this deficiency. So you need to know beriberi. Beriberi have 2 types, dry and wet. In the dry, you will have problems in the nerves and muscle. Nerves, the polyneuritis, and weak muscles. In the wet beriberi, you will have problems in the heart, okay, like heart failure, edema. The other one, Wernicke-Korsav syndrome, which occurs mostly in people who have excess alcohol consumption, they will have things like confusion, okay, mentally problem. So what happened is even if, for example, if someone is, for example, malnourished and you're trying to give them glucose, you're trying to give them food, they will not be able to observe it because the glucose goes into their body. The glucose is not able to metabolize because, as we have said, B1 is a very important cofactor. That's why you have to give them thiamine and glucose, okay? Now, B2 is bad. For the B2, it's very easy. You need to remember inflammation. So you can have inflammation in the lip and in the mouth. The inflammation that happens in the mouth, we call it stomatitis. And the one which happened in the lip, we call it glossitis. Okay? Remember the gloss that we use, the girls use it. So glossitis. And the mouth, stoma. So we have stomatitis. Okay. For the B3, niacin, you have to remember the 3 Ds. Okay. I love about B3 because it is B3. So also the disease which is associated, it will also have number 3 here. So the disease is about 3 Ds. So this disease, Pilagara the person who have this disease will be presented by these 3 things: dermatitis, diarrhea, and dementia. All of them starts with D. So yeah, they might bring it in the exam, all of this. For the coenzyme B5, there's no specific disease that you have to remember. You just need to remember that it's synthesized from pantothenic acid and it accepts and donates acid group. For the last one, which is a lipoic acid, which wasn't a vitamin, this is the most interesting for me, and I'm going to tell you why. This one can be inhibited by arsenic poison, okay? And so whenever someone has arsenic poisoning, what happens is that it goes to the lipoic acid. It's gonna stop the lipoic acid. It's gonna stop the cofactor, and thus the Krebs cycle will not be able to generate it. So the body will not be able to, to make energy, and this can actually lead to death. Like, this was a very famous poison used in the past. The very famous people who have been killed by this poison. Also, they also use it for in the The therapy. So they use it in the therapy to kill the cancer cells, not to kill the human. So not the poison itself, but the extract of it. And yeah, we can also use it in medicine as a drug, which help to kill the cancer cells. So the symptoms of this arsenic poisoning will be garlic breath, vomiting, diarrhea, coma, and death. Tamam? So yeah, that's it. Do you have any question? Okay. Do you mean the Krebs cycle or the diseases? Yeah, sure. Mm, Okay. Um, طيب. So we have, you can start memorizing it by the numbers. So we have B1. Give me a second. Yeah. 1, 2, 3. We don't have 4. 5, and then the lipoic acid. So B1, number 1, thiamine, it has got 2 diseases. So you can imagine it like this because this is the first one, B1. That's why it's not, this is not the reason. I'm just telling you, helping you to memorize it. So it will have 2 diseases, beriberi and Wernicke-Korsakoff syndrome. It happens in the malnourished people, okay? People who have malnourished or they abuse alcohol. Why? Because alcohol will impair the B1 absorption. Okay, like how can someone have deficiency in B1? Either by alcohol or by malnourished. They're not eating. They're poor, for example. It affects mainly the heart and nerves. You also need to remember the dry and wet beriberi. Like, yep, the wet one is basically gonna affect the heart. But the dry one is gonna affect the nerves and the muscle. Okay. For the other disease, the Wernicke-Korsakoff, this one will occur mainly in the people who abuse alcohol, and the symptoms will be confusion and alteration in the mental status. Okay. For B2, it's very easy. It's just the inflammation. Whenever you see itis, it's the inflammation. So it's inflammation, just as you see in the picture. It's the inflammation of the lip and mouth. That's it. خلاص. For the B3, as I've said, it's B3, and also the disease has 3 Ds. Okay, B3, NAD, 3D. That's how you can just memorize it. So we have got dermatitis. Dermatitis is inflammation of the skin, diarrhea, and dementia. Dementia means when you start to losing memory. Okay. Yeah. And then coenzyme A, B5, you don't really have a specific disease for this one. For the lipoic acid, which is not a vitamin, as I've said, it's not a vitamin, it's different. Another difference is that we have not talked about any disease. What the doctor talked about here is the poison, which is the arsenic poison. Okay. So this is gonna give the patient garlic breath, and the things like vomiting, diarrhea, coma. You can imagine it's the effect of the poison, and eventually death. Is it clear?
Yes, thank you.
You're welcome. Like now we have got 2 terms here to describe the TCA cycle. We have got first term which is amphibolic, and the second one which is anaplerotic. Amphibolic means that it's involved in both anabolic and catabolic process, and I think I have shown you how that we can link the Krebs cycle to other reactions. The other thing is that it's anaplerotic, which means that the reaction, it replenishes the intermediate of TCA cycle as they remove for synthesis of glucose, fatty acid, amino acid, or other compounds. So it's just the terms, like she might get it into an MCQ, asking you like she might just take out one word and like she might bring this term and ask you the definition or the opposite. طيب بسم الله. Okay. Now a very important thing is the regulation of PDH. If you remember that I have said this step, if you remember, this was the step before we started the Krebs cycle. So we had to generate acetyl-CoA, and after that, we generated citrate from acetyl-CoA, which went into the Krebs cycle. And we said it's a very important step because this is the one which is going to decide if we are going to inhibit or activate the Krebs cycle. Tamam? So now the things which are going to determine that are these 5 that I have written so far. So we have got acetyl-CoA, ADP, NAD+, calcium, NADH. So let me start with the first one. If we have so much acetyl-CoA in the body, do you think pyruvate dehydrogenase complex will be inhibited or activated? Can you please open mic if you're okay?
Activated?
Look, we are trying. Okay, now the purpose of this enzyme is to make acetyl-CoA, صح? Right?
Yeah.
Now imagine if we have so much acetyl-CoA, do we still need this enzyme to be activated and work hard?
No.
صح? So it will be inhibited.
Inhibited.
Yeah, it will be inhibited. Acetyl-CoA abundance or like so much acetyl-CoA is gonna inhibit it. طيب. What about ADP?
Yeah, I think we need PDH.
No, will ADP, if we had so much ADP, will it gonna inhibit or activate the PDH? Yeah.
Activate.
Yeah, because in this whole the cycle of the Krebs cycle, we actually wanted to make ATP, right? We wanted to make energy. So if we have so much, and ADP and ATP are always opposites, صح? So if we have so many ADP, that means that we don't have ATP, we don't have energy. So we want to make the energy. You can think about it like that. Okay. What about NAD plus or
Yeah.
NAD?
Positive.
Yeah,
Stimulates.
because the same thing, we're actually trying to make so much NADH, صح? So if we, in the cell, if we had so many NAD plus, this indicates that no, we have to work hard. We have to make NADH. So this is going to be activated. What about calcium?
No idea.
I'm going to give you a hint. The calcium is coming from the skeletal muscle. I don't know if you have taken a skeletal muscle.
We have.
Have you taken it? Okay. So what happens when you're exercising, when you're using your muscle, the muscles are gonna release the calcium in the cytoplasm, صح?
Yeah.
So in that
Activate.
time, exactly. You want to activate it because you want energy, you're working. What about NADH?
Inhibit.
Yeah, inhibit because because the purpose was to make NADH, and this one is suggesting that we have this one is suggesting that we already have so much NADH. خلاص, we don't need to activate it. Okay, now I was telling you activate, inhibiting, but how exactly this is happening, this is another story. Okay, so I know this looks complicated, but it's actually easy. Whenever you want to activate or inactivate PDH, you're gonna remove or put phosphate group, okay? So if you put Bismillah. If you put a phosphate group on the PDH, it means it's inactive. And if you remove it, it means it's active. And the enzyme which is going to do that is PDH kinase and PDH phosphatase. Is that clear?
Yeah, but
It's fine because it took me time to understand this concept.
I get that, but what's the full form of PDH?
Oh, it's pyruvate dehydrogenase complex.
Oh, okay.
Yeah. You saw we said that this is gonna inhibit it, this is gonna activate it.
Yeah.
So what do we mean? So we are basically saying that we have a phosphate group and we are trying to add it or attach it or detach it in a very simple way. So this is what's happening. So these are the things which are activating it, and these are the thing which is inactivating it. This is the little phosphate group which just goes. So yeah, that's all. If you have any question.
I have one question. Could you please
Sure.
repeat the full form for MCP, was it?
Oh, yeah. Um, mashallah, you remember the short form and wait, it was here. It's the mitochondrial pyruvate carrier. It's very easy, like it's telling you the function.
Yes, thank you so much.
Yeah, if you have any question, you can always email me. Uh, anything, not about this lecture, anything, you know, because I know first year is hard. It's not hard, it's more like you're new. You don't know how to study, you don't know how to organize your time, you're stressed easily.
Yeah, thank you so much. Wallah,
So yeah,
it was a helpful session.
alhamdulillah. Thank you so much for joining.
Thank you.
You're welcome.